phytochemical diversity and variation among germplasm, commercial lines, and is affected by planting season, ripening stage, and storage levels. And this work is, I will give it over introductions. However, for these, most of the topics will be the studies from the Gourd project, and these are the contributors. Some of them are sitting here. Dylan's here, it's our cucurbit breeder. Didi Landesma, our biometrician. And this is the groups from Chang'an University. So Professor Shao is sitting there. And also Mamta this morning, we heard her talk, and Warwick, and also Thomas. So I just listed outline, given the introductions, and then more focus on the studies, especially in the selected germplasm. We are not able to address all the germplasm, but it's selected. So these two, especially these two topics, I did not address the plant size. I think it's this morning we heard from Dylan, so this studies actually is also associated with what Dylan present. And some small topics about how the phytochemical, anti-diabetic phytochemicals that are affected by the ripening, storage, and planting seasons. Okay, so we all know that it's repeated information, but we know that we consume the vegetables, so it's very popular, especially in India, China, Philippines, Taiwan, Japan, and almost all over the world. And I also eat bitter in Germany when I visit in Germany. So it's really consumed worldwide, particularly it's in the Chinese and Indian communities. And also, bitter gourd consumed as a medicinal plant, and then it's well known that it's anti-hyperglycemia, and also anti-hyperlipidemia, and also anti-oxidations, anti-inflammation, antimicrobials, and also anti-cancer that we heard from Professor Paulenko this morning. The primary effects of the anti-diabetics, there are many effects, and also the bitter gourd, it's not, sometimes for some compounds that your biochemical pathway is quite straightforward. However, for bitter gourd, it's quite diverse. So for example, the primary effects of the bitter gourd, the anti-diabetics effect in the bitter gourd, it can be affects the plasma glucose, the homeostasis. So for example, that can decrease the carbohydrates, digestions, and also absorptions. And then also that can increase the insulin secretions. And also there is reports the data about increase the liver glucose utilization. And also can increase insulin sensitivity in liver and also in the muscle cells. They also have a minor or secondary effects of anti-diabetes. Reduce inflammation, and we know that inflammation is very important, the factors that might lead to the metabolic syndrome, metabolic disorders, and also antioxidants, and also reduce the delay of oxidative stress, increase the fatty acid utilization, lower blood lipids, reduce pre-fat cell proliferations, and also broad spectrum antimicrobial activities. And these are the effects that are all reported in the literature. So the focus will be in the anti-diabetic substances. So these are the more reported in the literature that I kind of try to categorize that the anti-diabetic substances into 4 major categories based on their chemical structure or properties. So first one, we normally from the water fraction is the protein related. So we've been hearing about the P insulin-like, and B insulin-like, lectin, and morphine, 1, 2, 3, and many others. And also the alkaloids, vicinine. And This is the major, also major groups that have been reported that have anti-diabetics phytochemicals in the bitter gourds. It has triterpenoid backbones. So these groups of the compounds that are listed here. So you can see that there's several compounds that have been very familiar, that have been repeated several times. And these are in the softening fractions. So in the morning we hear from Professor Colin Cole talk about the evaluations, the efficacy, and the of the different fractions. So actually from here we can see that phytochemical here from the softening fraction, from the water fraction, or overcrude extract, and also lipid fractions, they all have some anti-diabetics and also the reducing weight effect. For the lipids, the C9-C11 conjugated linoleic acid, also C9-T11-T13 conjugated linoleic acid, and other kind of fatty acids also reported to have the anti-diabetic effects. So in this study, we're more focused on the how to categorize the phytochemicals in the bitter gourd samples, and then I think I go to the introductions first. So here it's showing the structures that the triterpenoids compounds that you can see that they have these chemical backbones. They show the similarity, and this is cucurbitane type of steroid softenings. And then also they have a similar structure, and these are the fatty acids. So especially that this is the two double bonds here, and then C9 and T11 conjugated, and then there are also three double bonds, and then start from C9, T11, T13.
Oh, beautiful.
And also there are proteins, and some one other protein from the mammoding has already also the structure, protein structure, kind of categorized already, and then this is the from the smaller size, small peptides, and also this is the bigger protein. Okay. We've been hearing about the bitter gourd projects, but however, I have not have the opportunity to give some instructions. So with this opportunity, I would like to take a few minutes to introduce this project. It's overall it's a research project. However, we in the long-term goal, we like to address the to improve the income and quality of life of diabetes in developing countries. So with the specific objectives, we like to optimize the productions of anti-diabetes compounds in bitter gourd through varietal selection, post-harvest practices, and the food preparations methods. And then we also like to develop the evidence-based dietary strategies using bitter gourd to reduce hyperglycemia in type 2 diabetes population in Asia and Africa. We all know that actually bitter gourd is not new. I mean, it's been talking about that, and people are using it. However, when we talk about the dietary strategy or dietary recommendations, normally these kind of recommendations, we need to have evidence base. And sometimes we know that some variety is effective and some variety is not effective. And sometimes we get the samples get the fruits from the summer season, sometimes get winter season, and the effects are also all different. So in order to make it more clear, and then to have really make recommendations, use bitter gourd as the vegetables, I think we need more to generate more data in order to get the recommendations. So the overall this project approach a bit similar to the program today, the conference. We are kind of a look at the different aspects from germplasm to commercial variety, and then to field and the different location, harvest, post-harvest, and for preparation, and eventually we'll go to the animal and human studies. So for today, I will more reports from the study from here. And overall, that with this study, we hope that we can have a better, we can generate more evidence that would be able to make recommendations for production and also for consumption. And these projects have different, partners from over the world, and then several of them are sitting here. So basically, these projects implement in AVRDC headquarters in Taiwan, and also in AVRDC. Not changed the name already. East and South. Okay, the office in Bangkok is here, and also the offices in Hyderabad, and then we also have the partner from PAU and partner from KCMC in Tanzania, and also from AVRDC RCA, the Regional Center for East Africa, and also we have partner from Germany. And this is 3 years ago, we have the project workshops at the AVRDC headquarters. Okay, so go to the study. We would like to have the germplasm evaluations that this morning, Dylan have already reported the horticulture data, and then he's hoping that I can tell more about the correlation. But here I would like to say that because a lot of analysis is just finished not long ago, so today what I report will be the preliminary analysis, statistical analysis. So a lot of still we need to make many correlations. However, today we'll not be able to report all the correlations in terms of the phytochemical and also horticulture traits. The study design, it's RCBD, 3 replications, 8 plants per replications and 2 years. And then we have the horticulture traits and also the anti-diabetic compounds, and then through the LC-MS profiling. This study conducted at AVRDC headquarters in Taiwan. So first, we have, because we just see that there are many compounds that involved in the anti-diabetic effects. From the nutrition point of view, sometimes we need one items we will need to involve in one type of analysis. So then we think that how that we can just our previous speaker, I'm talking about the one HPLC, maybe you can show several kinds of pigs, and this is the similar approach. We're using the global profiling approach, and at first using untargeted, and later on that we tried that we can use one extractions, then can catch as much as possible the compounds. However, this is not possible. One extraction can have all the compounds you catched. So for example, that we use 80% methanol to do the extraction, that actually have already determined what kind of compounds in it and what kind of compounds will be excluded there. So this kind of extraction will not be able to include the protein here. But probably will capture the softening fractions and also the lipid fractions, part of the lipid fractions. So these are the compound identifications were contributed from the Tonga University, from Professor Shao's group. So these are the profile, the LC-MS profiles from one of the beta Gourd samples, and then total 16 compounds so far is identified, and more compounds will be identified from the LC-MS profile, and that includes 7 phosphotylcholines and 2 fatty acid derivatives, and also 5 phytopenoids, 1 alkaloids, and 1 amino acid. For today's presentation, because there are many types of phytochemical, which I try to group them in the 3, so that it won't be too complicated to compare the results. So I grouped the fatty acid into PICS group 1, and then I grouped the 5 tryptopanoids in the PIC group 2, and then the alkaloids in the PIC group 3. And so far I did not include the amino acids. So here's the compounds, and even I did not include all the fatty acids. I only include the linoleic acids related fatty acids. And these are the 5 tripeptanoids that identified, and I include all the 5 one. And this is the one alkaloids, this thing that we have already identified. So actually only one compound in the group 3. Here just kind of repeated again that we actually catch part of the lipids as the group 1, and then the triterpenoids in the group 2. However, we did not include everything, and we have not identified everything. And then this is alkaloids as the group 3. We are not able to include the protein fractions. Okay, so for the germplasm trials, that this is the group means, and then this is 2001 planting season. The samples from 2001 planting season, and this is the second year seasons. Here it's only the, I have not made the concentrations, but here I use the LC-mass intensity. Okay. So we can see that from the year 1 and year 2, the year 2 slightly increased for the group 1, that's fatty acid. For the group 2, also slightly increased. For the group 3, it's alkaloids, and also slightly increased. And then we also can see the compound intensity range. For the fatty acid group 1, 30 to 60. The second year is 30 to 68. It's quite stable. For the group 2, it's 0.2 to 9.4, and the second year is 0.2 to 13.9. And then this is the group 3, 29 to 95, 24 to 103. And we evaluate the 53 sessions in the first year and second year, because it's really very big trials. And the second year we narrow down to the 25 sessions. So here is the analysis based on the 25 germplasm sessions that Dylan have already presented. That the 25 we did not select just this similar type of fruits. We select the most diverse groups of the bitter gourds. So here you can see I just simply put the highest value, the top 3 highest value, and the lowest value of the 3 varieties, 3 germplasms. And then we can see that for the group 1 fatty acid, here is the repeated germplasm sessions for the year 1 and year 2. That means it's more stable for these sessions that the compounds in the year 1 and year 2, they keep the same ranking. However, in the for the triterpenoids, that the compounds range, the session range ranking are different. So that means it's more diverse. It's not so stable. And then the group 3, we can see that the most stable group from this germplasm, we can see it's the alkaloids. So it's not much affected by the year factor. And the second trials, we have the commercial variety multi-locational trials. We include 20 commercial lines that from seed companies that Dylan reported this morning. And then we have the RCBD design and 3 replications, and for 2 years. Want to see the genotype environment interactions. We conduct this in the 4 locations, Shanghai, Taiwan. This is where that AVRDC headquarters are located. South India, this is AVRDC in Hyderabad, and also North India at Punjab Agricultural University, and Tanzania. So we have the horticulture and also the anti-diabetes evaluations. One of the questions raised, because we want to have a multi-locational trials, and then also we want to compare the phytochemicals from the different locations. We first have to think about the how that we can get the samples that is comparable. And so far, before the laboratory analysis, the freeze drying probably is more commonly used to keep most of the phytochemical or nutrients compounds more stable. However, this is not feasible in the locations, so that we also try that how can we compare the freeze drying and also the open air drying. So that because most of sample from the regions will be sent as a oven drying sample compared to the samples at the headquarters. So at the headquarters, Taiwan AVRDC headquarters, we have the freeze drying and the oven drying. And from the ANOVA analysis, we can see that there is significant effects in the replications and also variety and also drying. However, there is no interactions of the variety and drying. So drying effects, it's more depends on the chemical effects. It's not depends on the variety. So when we compare the 2 years, 2011 and 2012, for the freeze drying samples, we can see that this is only in Taiwan, one of the locations. Then we can see that the 2012 year, then the compounds intensity reduced. And when we compare about the freeze drying and oven drying, the oven dryings that even intensity also reduced. So eventually, when we compare the regional data sample from regional from the regions that we use the oven drying sample. However, we can see that the oven drying and freeze drying is not affect the compounds in the group 2 and in the group 3. It's not affected. The group 2 kind of at the same similar level, and the group 3 it's more affected by the ears. It's not have the same trends like the fatty acids. So in terms of the chemical properties, the triterpenoids and alkaloids actually it's quite stable compared to the other nutrient compounds. So this is the same things I represent the same way. The content range or intensity range for the different compound group, and also the highest 3 variety and then the lowest 3 varieties. And the red color indicates the repeated in the two years. So we can see that the content range, these two are slightly different, and the other is similar, and some years is a bit higher, and then the second year is lower. However, when we compare the freeze drying and the oven drying, then you can see the content range, it's not different much for the group 2 and group 3. And I think I'm not going through all the names, but just like you to see that how many red colors, that means actually for the group 2 and group 3, it's quite stable in terms of the compounds. They are not jumping around. And this is the results from the PAU, the North India. Here we can see very clear the yearly effect, and also the content range it's lower than the AVRDC, the headquarter. And here we didn't see the red color. That means the ranking actually it's all different. We still need to try to figure out why the concentration is the whole the phytochemical concentration is lower from these regions. This is the study from Hyderabad, South India, and the fruits produced a similar content range like the samples from AVRDC headquarters. Though the rankings are a little bit different from the headquarters, but we also can see that for the group 2 and group 3, the ranking is more stable. And this is the data from Tanzania. Also, we hear that the yield is pretty high in Tanzania. There is a great potential to grow bitter gourd in Arusha, Tanzania. And also the phytochemical range is as high as the data from Taiwan. However, we only have the 1 year data. I just simply put all this content range together so you can easy to see how that can compare. So germplasm, we apply just the freeze drying. So this is more, so the drying process, the compounds we hope is not affected by the sample preparation methods. And then the commercial, we have the freeze drying and over drying. And actually that we see that for the group 1 sample, the over drying affects, reduce the compounds in group 1, that is fatty acid. However, for the group 2 and group 3, it's not much affected. So we use this data to compare the other regions, and then we can see that actually the phytochemical contents at headquarters and also from South India, that's AVRDC Hyderabad, it's quite similar. However, from North India, it's several times lower. And this is also quite comparative among the 3 regions in terms of the compound range content. Okay, a small post-harvest study that we look at the 3 harvest days, that is the day after flowering. For the all the fruits that we get, it's time. The fruit that we get, it's the marketable stage. Marketable stage means that when it's the green, so it's not a ripened, not the yellow one. Okay, and then slightly different the 3 days after flower. So it's not too much different because at this study that we did not try to evaluate the not marketable stage. Okay, and also 2 drying method that I just explained already, and 3 storage lengths, that is fresh and 1 day and 2 days storage. So I think because running out of time, I just simply give the conclusions. And the way that I present the data is similar. And I use one example. It's the NS1020 as the example in the study. Okay, because of the times, we also look at the dry matter, vitamin C, carotenoids, antioxidants, and this is storage from year 1 to 1 day, fresh 1 day, 2 day, fresh 1 day, 2 day. So you can see carotenoids increasing. And also that we also look at the compound affected by planting seasons. So we look at the 2 variety, green and white. Study design RCBD3 replication, 2 years, 3 seasons. And we use the LC mass, UPLC 12 mass, and for the analysis method, this is conducted by Professor Shao's groups. They're using the pattern recognitions. So that actually after the LC mass analysis, use the PCA analysis that we can group the sample, bitter gourd sample by different seasons in 1 year. It's very clear. And based on these samples, then we can identify the most affected metabolites in bitter gourd affected by the seasons of the year. So these are the, I didn't list the 30, but these are the samples belong to amino acid, alkaloids, fatty acids, lysophosphatidylcholines and isophosphatidylethanols and triterpenoids. And we've been hear this again and again, and these are also the major compounds that are found in the bitter gourds. Okay, go to the conclusions very fast. So this is the conclusion from the study only, and then preliminary conclusions based on the preliminary analysis. So the varietal differences were found for all 3 compounds groups. And among the germplasms, the variation within fatty acid, triterpenoids, and alkaloids were 4-fold, 10 to 7-fold, and 3-fold respectively. So the trends of the variation within these 3 groups of compounds were similar for the commercial varieties. So there are opportunities exist to improve the all three compounds. However, this is in terms of the variations, not in terms of a metabolic pathway. Sometimes it is also different. A longer storage can lead to decreased fatty acid and vitamin C content and increased the carotenoids, antioxidant activities, but not for the triterpenoids and alkaloids. And metabolite types and amounts in the bitter gourds were strongly affected by the seasons. So the 30 most affected metabolites were identified that belong to the groups I just read. So the most affected is amino acid, and then alkaloids, fatty acid, and also the other groups. For this, thank you very much for listening.
Maybe we can have just one question. So no questions. We thank you very much for your very informative and elaborate presentation on anti-diabetes.